growth. So because of that, usually you may add a preservative to a syrup. Okay, we looked at redox potential, and we said that with these microorganisms, usually you want a terminal electron donor. Usually after most of these respiratory reactions have happened in an organism, you end up with free radicals, okay? Some of these free radicals need a terminal electron donor so that they do not accumulate. But if you don't get a terminal electron donor, they tend to accumulate and therefore they will kill the microorganism. Okay. Then we also looked at pH, the importance of pH, and the fact that microorganisms will tend to grow at the neutral pH. Okay. Extremes of pH may not be favored except for some unique microorganisms. Okay. Packaging design, again, we looked at that. Protection within the pharmaceutical product, we say that some substances within your product may actually act to shield your microorganism from destruction by either processes, sterilization processes, or even okay. Then we looked at the type and size of contaminant, and we said some contaminants are very virulent. You get a small amount into your product and it generally just causes destruction. I mean, they multiply, okay? And we said that some of them can actually be dealt with by the preservative system. Then we also looked at the size of the contaminant, and we said if you have a very large contaminant size or inoculum, what will happen is the large amount will tend to interfere with the overwhelm rather. It will overwhelm the preservative system. Okay, so because of that, usually you want to avoid large contaminant inoculum. Okay, and that's why you will control contamination, for instance, in raw materials, in water, in all that. Okay, you limit the contamination, because remember, if you have a very high contamination, then probably the method you're using may not be adequate to adequately destroy the microorganisms. Okay, so as we start off today looking at preservation, first I want us to look at what are some of the causes of a high contaminant inoculum. Okay, what would cause your product to actually get a high inoculum of contaminant? Okay, so the first one is if your raw materials are unusually contaminated, okay, that would actually cause a high contaminant inoculum, and that is why we control this contaminant, okay, so that we don't have a high, unusually high, because remember, one of the things hopefully you were told during your dosage form, okay, or probably let me mention it now if you are not told, is sometimes depending on the kind of product that you're producing, sometimes you may be allowed a certain limit of microorganism, even when you're starting with you're preparing what are they called, sterile products. Okay, you may be allowed a certain limit of microorganism in your raw material if that product is going to undergo terminal sterilization, or if at some point during the processing of that product there will be a process that will ensure removal of those microorganisms. Okay, you will be allowed to actually have a certain limit. Okay, so but the limit is so that you don't have start with materials that are unusually contaminated, because if the contamination is too high, then usually what tends to happen is that the microorganisms will overwhelm the processing, okay, or even the preservative, okay. So you could also, another cause of high contamination is delay or failure in the plant cleaning protocol occurred. Remember we said before you start producing another product, you have to ensure that cleaning is done and line clearance is done, so that you are able to prove that the previous product has been uh, everything related to the previous product has been removed from the production area. The machines have been cleaned, okay, and QA has approved starting a new product, okay. That is the process involved with line clearance, okay. So if should you get a scenario whereby you have a delay or failure in plant cleaning and protocol, then it may actually encourage high contamination. Okay. Then you can have scenarios whereby large microbial growths detached themselves from within supplying pipework. Again, think of it this way: you have pipework or pipes that are supplying water, for example. Okay. Now, as this pipe supply water, it's coming in through the pipes, okay. Expectation is that there will be the pipes are clean, okay. They are not allowing any microorganisms to go through. And usually one of the things that you will, especially for those of you who went to the larger industries, if you are keen, uh, you hopefully were told that they don't usually allow water to stagnate in the pipe, okay. There is always water flow from the uh, storage tank through the pipe at all times, okay. And usually the water flowing through is at a certain temperature, okay. And this is because if you leave the water to stagnate for some time, you may find that if you had microorganisms in that water, it's possible for the microorganism to colonize an area within the pipework, okay, and then multiply, okay, so that next time when you allow the water to flow, that colony detaches, okay, and it's possible for it to end up in your product, okay. So that's what we are talking about here. Large microbial growths detach themselves from within supplying pipework, okay. So some of these huge companies will ensure that there's always water flowing, so that at no point will you have a dead leg, such that in case of colonization, now the microorganisms are able to detach and contaminate. Then a change in production procedures allowed unexpected growth of contaminants during the modified operation. Okay. So again, remember we talked about, okay, someone will be handling validation, but even earlier it has been alluded to, okay, that when you come up with a production procedure, you have to validate and ensure that the procedure consistently gives you the expected results, okay. Now, if you change a procedure, okay, without validation, it's possible for you to actually get contamination. That's what we are saying, okay. Another cause of high contamination, there could have been destructive work in the vicinity of the manufacturing site. Remember, we said maintenance should not be done uh, when production is going on. Okay. because if this happens, then it's possible for you to end up with contamination. Okay. There have been also, it's possible again, another cause of high contamination is when there had been gross misuse of the product during administration. Again, think of something like an eye drop. Okay. If you have an eye drop and this is supposed to be sterile, okay, and usually you'll be told to, as you're using it, ensure the tip does not touch, the tip of the dropper does not touch any part of your skin, any surface, okay? Because if it does that, and don't touch the tip with your bare hands, and again, before you start instilling the drops into your eye, ensure that you have washed your hands are clean, okay? Now, think of a patient who does not follow those instructions, okay? They come to put the eye drop in their eyes with dirty hands. They touch the tip of the eye drop with dirty hands. The bottle falls down. I mean, all these are scenarios where the eye drop is actually able to collect germs, okay, which may end up in the eye. That is what we are talking about by gross misuse of the product during administration, okay? Instead of keeping the product sterile, ensuring that sterility is maintained, the patient is not keeping it sterile. Okay, you look at the bottle and you can just see this patient came with dirty hands, touched the bottle, so you can imagine because of that, microorganisms have been introduced into the product. Okay, so these are some of the causes of high contaminant inoculum. You can think of many others, okay? Just scenarios where you end up with more than a high load of contaminant. And the problem with this is that if you're using a preservative, this high load may overwhelm the preservative, okay? Or if at some point during the production you're expecting that the microorganisms may be killed because of the load, it may also overwhelm the process, okay? So let's go into preservation. This is carried out using antimicrobial agents. Preservation is the process of inhibiting or minimizing, preferably during storage and multi-dose application, the risk of microbial contamination of pharmaceutical products. So again, remember last week, I think I mentioned this, that uh, you're not adding preservative. Here we have said we use antimicrobial agents as preservatives or for preservation. But keep in mind, you're not adding a preservative to take care of inadequacies in your production, okay? Your production process should be uh, very precise to so that you do not introduce any microorganisms during production. So we are not adding a preservative to take care of inadequacies during production, okay? Our understanding is that at the end of production, our product is good, okay? But we know that during storage and multi-dose application, it's possible that you may get introduction of microorganisms, okay? So to avoid the microorganisms or prevent the microorganisms from thriving, we add a preservative, not for inadequacies during production, but so that in case there is introduction of microorganisms during storage or multi-dose application, then we uh, reduce the risk of contamination of pharmaceutical products. Okay, when we talk about multi-dose applications, what comes to mind? Tabitha, when we talk about multi-dose application, what comes to mind? Yes. Yes, so that is multi-dose application, okay? Like the case where Tabitha has told us with the vial, okay? Again, if you think of eye drops, okay? Again, you have multi-dose application, okay? Because you will take the eye drop, instill in the morning, okay? However, number of times you're instilling in a day, okay? Same eye drop, instill in the morning, come at lunch, 8 hours later, instill, 8 hours later, instill, okay? So that means that there are several doses in that product, okay? And it will have to be accessed a number of times. So we don't want a scenario whereby when it is accessed, it is actually uh, contaminating the product. Okay. So some of the major preservatives include citric acid, benzalkonium chloride, benzoic acid, salicylic acid, phenolic compounds, among others. Okay. There are quite a number that you'll come across in practice. Okay. Preservatives are included in a formulation to further reduce the risk of spoilage and kill any anticipated low levels of contaminants remaining in a non-sterile medicine after manufacture or which might enter while stored, okay? Or during the repeated withdrawal of doses from a multi-dose container. If a medicine is unlikely to encourage growth or survival of contaminants and infective risk is low, such as with tablets, capsules, and dry powders, then a preservative might be pointless. Okay? Preservatives should, however, not be added to deal with erratic failures in poorly controlled manufacturing processes. So, again, this is basically what we have said. If you have a pharmaceutical product that naturally, when well produced, will not promote growth of microorganisms, then there is no need to add a preservative to it, okay? Because if you think of a tablet, for example, if it is well dried and well packaged, ideally there should be no growth of microorganisms because to begin with, it does not have enough moisture to facilitate growth of microorganisms. But if you did not dry it correctly, then it's possible for it to be able to facilitate growth of microorganisms. Okay, so if we see you adding a preservative to a tablet, then we might actually question your processes. Okay, because maybe you're actually just doing it to, cover this last statement we're talking about here, to deal with erratic failures in poorly controlled manufacturing processes. And we have said you do not use a preservative to cover uh, shoddy work. Okay. Products that are preserved, we have pharmaceutical products, we have medical devices, we have cosmetics, we have dental products, okay? And there are examples over there. So these are basically, you don't want, these are used, okay, medically, and you do not want to introduce microorganisms into the human body. Okay, so all these are actually preserved to ensure that the product is maintained at a safe, or rather in a safe state, to ensure that uh, the patient or the user is protected. Okay. So some of the factors that affect preservative activity in a pharmaceutical product, I want us to look at those. The first one is interaction with formulation components. Then you have properties of the preservative. Uh, you have effect of the container. Okay, and here we are looking at glass, we're looking at plastic, we're looking at rubber, we're looking at corks. We have the type of organism or contaminant, okay? We have pH. These are factors that will affect preservative activity. What are we saying? If you have a product and you add a preservative to it, okay, you expect that the preservative will stop the growth of microorganisms, okay? Now, what we are asking is if it is not able to stop the growth of these microorganisms, okay, what could be the reason? Okay, could it be that the preservative is interacting with other formulations in your product so that the preservative is not available? Remember, one of the things I should mention at this point is that only the free preservative is able to stop uh, growth of microorganism. Okay, if you have a preservative that is not free, then it will not be available to stop growth of microorganisms. So we are very concerned with the free preservative. So that in this case, if you get interaction of a preservative with formulation components, then what will happen is that you will not have free preservative. Okay, as much as the preservative, yes, is in your product, it is not able to stop microorganisms. So think of it this way: you have a bottle of syrup. Okay, think of a bottle of syrup, and you place a preservative or a solution. Okay, you place a preservative in there. You've calculated the correct quantity, the correct that will give you the correct concentration. You add the quantity into the bottle. Okay, but the preservative gets into the bottle, but now it interacts with a component in the liquid. Okay, so now you end up with having you don't have free preservative because the preservative has interacted with a component. Okay, so what happens? It means that if you introduce microorganisms into that product, they will grow. Okay, and they are growing in the presence of preservative because the preservative is not free. That's why we are saying you must have free preservative. Now, the disadvantage of this, or one of the issues with this, is that as much as the preservative is uh, not free and therefore not preventing growth of microorganism, it is able to contribute to the toxicity. Okay, because it is there, it will end up in the body of the patient. Okay, if you increase the amount beyond the toxic levels, then this will affect the patient. Okay, it is able to cause toxicity to the patient, even though it is not participating in preventing growth of microorganisms. Okay, so that's why it's important for us to look at these factors affecting preservative activity, because we can't just indefinitely increase the amount of preservative. We will hit toxicity level and we will actually affect our patients. So let's start with interaction with formulation components. Most preservatives interact with many of the ingredients or excipients of formulations via a number of weak bonding attractions, as well as with microorganisms present. So again, remember we've said only the free preservative is effective against microorganisms. Okay, so if your preservative is interacting with excipients, it means that it is not free, therefore microorganisms can grow. Okay, so this can result in unstable equilibrium in which only a small proportion of the total preservative present is available to inactivate microbial mass. The unavailable preservative may still, however, contribute to the general irritancy, and at this is the toxicity we are talking about, that the preservative will contribute to the general toxicity of the product. Okay, because if you add more preservative, it means that the preservative will end up being toxic. Okay, just because it's not effective on the microorganism doesn't mean that it is not affecting the patient. It is still affecting the patient. Okay, where the solute concentrations are very high and water activity is appreciably reduced, the efficiency of preservatives is often appreciably reduced and may be virtually inactive at very low water activity. Okay, so again, uh, the preservatives usually require uh, enough water activity. Okay, I mean, the solute concentration, like in the case of the tablets, remember we say uh, if you have a tablet, for example, that has a low water concentration, okay, we said that the tablet will not allow growth of microorganisms, okay? So there's no need to add a preservative to that. But in addition to that, even the preservative themselves, usually they will require a certain amount of water activity for them to be active. So if you're adding a preservative to a tablet, okay, and yet the tablet does not have enough water activity to facilitate growth of microorganism, neither does it have enough activity to facilitate activity of the preservative, and yet that preservative is contributing to the toxicity of the product. You see now you're actually tripping. You're not adding value to the patient, okay? So it doesn't make sense to add a preservative to a low water activity product, okay? Therefore, the practice of including preservatives in low water activity products, such as tablets and capsules, misses the point, okay? Another factor that needs to be considered is the effect of the product pH. Okay. In weak acidic preservatives, activity resides primarily in the anionized molecules, and they only have significant activity at a pH where ionization is low. Again, remember from your biochemistry, pKa, okay, of a product will determine the percentage of the product that is ionized at a specific pH, okay? So later on, we'll be looking at different preservatives, types of preservatives. And one of the things you'll be able to appreciate, there's a group that consists of acids, okay? Now, this include things like benzoic acid and sorbic acid, okay, which have a pH of 4.2 for benzoic acid, 4.75 for sorbic acid, okay. These have a limited preservative usefulness above a pH of 5, okay. And this is because above the pH of 5, most of these preservatives are actually ionized, okay? And one of the things that you'll realize is that when you look at the cell membrane is charged, okay? And generally it will tend to repel charged charged molecules, okay? And that's why on the cell you have channels, okay, that will, if for some of these molecules to go through that the cell needs that are charged, you will find channels that and transporters that will facilitate entry of these products, okay? But coming back to the case of benzoic and sorbic acid, generally acids, okay? If the acid is predominantly ionized, then usually what will happen is because it has become ionized, it will no longer be able to cross the membrane, okay? So it cannot enter the cell, okay? That means that if your product is above a pH of 5 and you're using benzoic or sorbic acid, it will not be effective because at the point at which it is getting to the microorganism, it is ionized. Therefore, it will not be able to enter the microorganism. Okay, you need it to be non-ionized for it to be able to penetrate the microorganism. Then after it has penetrated, it gets into the cell, finds a higher pH, becomes ionized and interferes with the activity of the microorganism, okay? But if it ionizes outside the cell, it will not be able to access the cell, okay? So if you think of parabens, the parahydroxybenzoate esters, they have a non-ionizable ester group, okay? and a poorly ionizable hydroxyl substituent, okay? And their pKa is around 8.5, okay? Therefore, because of that, they have a moderate protective effect even at neutral pH levels. So the good thing with these parabens or parahydroxybenzoate esters, they are, by the time they are getting access to the microorganisms, they are not ionized, okay? Because they have a very high uh, pKa, okay? So they are able to penetrate the cell and destroy the microorganism, okay? The activity of quaternary ammonium preservatives and chlorhexidine probably resides in their cations. They are effective in products of neutral pH, okay? So again, for these quaternary ammonium preservatives at higher neutral or higher pH, they are actually ionized, okay? So they are in their cation form. And the beauty is, this is the form at which they are active, okay? So again, which makes them effective. Formulation pH also directly influences sensitivity of microorganisms to preservatives, since microorganisms are susceptible to extremes of pH. Again, we had mentioned that, okay? Another one is the efficiency in multi-phase systems. Now, again, remember, when you think about multi-phase systems, think of systems like oil in water, okay? Emulsions. You have oil, then you have water part, okay? Or a water in oil emulsion. Basically, you have two phases. You have the aqueous phase, and then you have the oil phase, okay? Excuse me. Now, with the different preservatives, what you will find is that they will have solubility in different phases, okay? So you can have a preservative that is soluble in water, okay? Or you have a preservative that is soluble in oil, okay? What happens is that if you have a preservative that is preferentially soluble in water, what will happen is that uh, you will get growth of, or rather, you will get the preservative being partitioned in the water, okay, than in the oil, okay, which is a good thing, because one of the things you will find is that microorganisms generally, the microorganisms of importance, the ones that we are concerned about, generally will multiply in the presence of um, the aqueous phase, okay? So you want a preservative that is preferentially partitioned in the aqueous or water phase. If you get a scenario whereby you have a preservative partitioned in the oil phase, then what is going to happen is your pharmaceutical product will get infested, okay, or rather it will get colonized by microorganisms. Reason being, the microorganisms we are concerned about will tend to thrive in water. Okay, so if your product or if your preservative is soluble in oil, okay, it means that most of the preservative will end up in the oil, then your water phase will remain without preservative. What happens? Microorganisms will grow in your water phase and destroy your product. Okay? So that's why we are saying in a multi-phase formulation such as oil in water emulsion, preservative molecules will distribute themselves in an unstable equilibrium between the bulk aqueous phase and the oil phase by partition. Okay? Another multi-phase system is a system that contains surfactant micelles. Okay? If you have a preservative and it gets sequestered within a micelle, okay? I hope you remember what micelles are. We did this, we learned about this in third year. Okay, so remember we agreed that we are leaving nothing behind. So we looked at micelles and we said that micelles can be used to solubilize, okay, now pharmaceutical product. The other thing that you need to note is that they may not only solubilize the pharmaceutical product, okay? Remember, micelles will offer a hydrocarbon center, okay? Do you remember I hope you remember the structure of a micelle? Okay, let's see if you remember the structure of a micelle. Walter, can you remind us the structure of a micelle?
Hello, good morning.
Good morning.
Yeah, so the structure of my cell contains a hydrophilic head and a hydrophobic tail.
Mm-hmm.
It looks like a little round ball with the tails sort of forming a circle outside, and the tail and the hydrophobic tails sort of coming to the center to trap or to create the micelle.
Okay. Yes. Okay. So from what, and let me just share my screen to remind us a bit of what Walter is talking about. This is from the third year lecture. Okay. So this is what he's talking about. You remember the hydrophilic head and hydrophobic tail. Okay. When if you add micelles to a substance like water, usually what will happen, the micelles will tend to go to the surface, okay? They reduce the surface tension at the surface, and then when the surface is full, they self-associate, so that now these molecules come together to give you a ball-like structure, okay? This is like a ball, and you have the heads in contact with the water, okay? And then the tails give you a hydrophobic center, okay? A hydrophobic center that behaves like a hydrocarbon. So outside here, you have water, but inside here, it behaves like a hydrocarbon. It's hydrophobic, okay? So if you have oil, something oil, it will come into the center of the micelle. Or if you have a substance that is more hydrophobic, okay, it will come into the center of the micelle. That's why for hydrophobic substances, okay, when we want to solubilize uh, hydrophobic APIs, you can use micelles and they will come and get solubilized in here. And then because now they're in liquid form, it's possible for them to get absorbed. Remember we said for absorption to occur, your API must be in solution, okay? So it's possible for it to get solubilized in here. Now, it's not only the API that can get solubilized in here, okay? Any other substance, okay, that preferentially prefers hydrophobic uh, properties or environment can also get solubilized in here, okay? So that's why we are saying that if you have a preservative, okay, in a product that has micelles, it's possible for the preservative to actually get sequestered within the micelle. Now, the problem with the preservative getting sequestered within the micelle is that for the preservative to be active, it must be free. But now, if it is sequestered within the micelle, it is not free. Okay? So what that means is that now you do not have free preservative, your pharmaceutical product is not well preserved. So the implication is that you may actually end up getting a spoiled product because it is going to be colonized. Then we have other scenarios where polymeric suspending agents and other solutes by competitive displacement of water of solvation. This one will actually get sequestered in your polymeric suspending agent, okay? Particulate and container surfaces by adsorption. It gets adsorbed at the interface, at the surface, okay? And then it can also get absorbed by microorganisms, okay? Although this is actually more adsorption rather than sequestration within a phase, okay? So the key points with the multi-phase systems, we are basically looking at partition and solubilization, okay? But again, if you look at, this solid as a phase, okay, then it's possible you can include it there, okay? Where you have a liquid phase and a solid phase, therefore there is adsorption onto the container, okay? So the overall preservative efficacy can be related to the small proportion of preservative molecules remaining unbound in the bulk aqueous phase. So remember we've said it's only the unbound preservative that is effective, okay? So if it is, some of it is getting sequestered in the micelle or in the oil phase, it means that only a small amount is remaining active. Therefore, you may not have preservation in your product, okay? So although as this becomes depleted, some slow re-equilibration between the common components can be anticipated. So again, remember we said there's equilibrium. There's an equilibrium where you have preservative that is free, okay, and preservative that is sequestered within the micelle, okay. So because of that equilibrium, if you increase the amount of preservative, okay, then it's possible for you to actually ensure that the micelles are actually uh, fully occupied, then you have free preservative. So it's possible for you to add more preservative. For instance, if you think of the oil phase, okay, we are talking about partitioning. There is a partition coefficient whereby a certain proportion of the preservative will end up in the oil phase, okay, and a certain proportion of the preservative will be free, okay. So if you want to increase the amount of preservative, it's possible for you to achieve the required concentration that is able to prevent growth of microorganisms. However, remember we said that this preservative, as much as it is sequestered and is not contributing to preventing growth of microorganism, it is present in your product and is contributing to the toxicity of your microorganism or of your product, okay? So if you add more preservative to it, it will end up becoming toxic, even though at some point you will achieve the effective concentration that is able to stop multiplication, okay? So you have to be careful. In some cases, you can add, if the partition coefficient is able to allow you to add preservative, you can add, so long as you do not exceed the toxicity level, okay? If adding more preservative is going to cause you to exceed the toxicity level, then you are not allowed to add. You better change the preservative and pick something that is more compatible with your product, okay? So the loss of neutral molecules into oil and micellar phases may be favored over ionized species. Again, think of the products that tend to ionize. Okay, so if you have a product that is unionized, okay, then chances are it will tend to move into the oil phase and the micellar phase. Okay, the ionized species probably may not end up in the uh, ionized, or rather in the oil and micellar phases. Okay, although considerable variation in distribution is found between different systems. So again, it's just important Then we have effect of container or packaging. This is another factor affecting preservative activity. So preservative availability may be appreciably reduced by interaction with packaging material. Okay. So for instance, we have permeation of phenolic preservatives into the rubber wards, and teats of multi-dose injection or eye drop containers, and by their interaction with flexible nylon tubes for creams. Okay, so if you have a product where you're using, if you have a product where you're using a container that has rubber, okay, it's important for you to appreciate that some preservatives may get absorbed onto the rubber. And if it gets absorbed onto the rubber, what can happen is that, okay, can you guys hear me? Okay. I I can see a number of you can hear me. I'm wondering why the class suddenly is half. Okay. There's a bit of breaking.